So it's our real privilege and honor to have Barry Connors here as our first external speaker for the GT Neuro Seminar Series. So Barry did his PhD at Duke University and then did his postdoc at Stanford and had his first assistant professorship, I just learned this morning, at Stanford. And thereafter he moved to Brown, and he's been at Brown since the early 80s for the rest of his academic career so far. He's been the chair of the Department of Neuroscience there for the last 10 years or so, right? In addition to
and
that,
not for much longer either. End of this year, I'm done.
okay, so only this year. This is the last of the great 10 years. So he has been extremely active. If any of you have sent papers to the Journal of Neuroscience in the last 10 years or so, he's been active at the highest levels as editor of the Journal of Neuroscience, also cerebral cortex, and he's been active in study sections in a variety of capacities for the NIH over the last 10, 15 years as well. He's also been cited for his enthusiasm for undergraduate education. He's written textbooks for neuroscience, basic neuroscience, and he's also been cited for his excellence in mentorship by some of his postdocs and grad students. So he's really a well-rounded example of a neuroscientist, and it's a good thing for the students to talk to them about mentorship and choosing a good mentor for your postdoc. So in terms of science, if any of you have cited anything about the cortex and studies in vitro and slices, if you trace it all the way back, it goes all the way back to Barry. He did some of the first recordings of cortical cells in slices and identified things that we take for granted now, things like bursting in layer 5, how bursting responses can give rise to pathological rhythms such as epilepsy, another great interest of his. And he's also developed very powerful preparations for looking at how thalamus and cortex communicate with each other in vitro and slices mechanistically. And I think this is what he's going to tell us about today. So again, we're very welcome to have you here. Thank you.
Thank you.
Yeah, okay. Just wave your hand if fade away.
To do. Anyway, it's great to be here. Thank you for the kind introduction, overly kind. My first trip to Georgia Tech. I understand this is being simulcast at Emory, so to the person at Emory, you know, I'm glad to be there too. What I'm going to try and do today is tell you 3 stories. And actually, before I even start, is there any way to dim the front lights? Because there's a lot of fluorescent things here that might show up better, but it's not a big deal. But yeah, okay. It's not. We'll just press on. What I'm going to do is I'm going to tell you some work that we've been working about an issue that we've been working on for quite a few years now. In fact, since I was a postdoc, if I'm truthful about it, how the thalamus and the cortex interact with one another. And in particular, I'm going to focus on the way the cortex interacts with the thalamus. And thanks. That's good. So I'm going to, first of all, give you a bit of an introduction, tell you about where the thalamus is. That red part there, just to orient you, is your thalamus in the humans. I'm not going to talk about the human thalamus. I'm going to talk about mice. Cortex is in brown here, but that's the basic gross anatomy of the system. And in particular, I'm going to focus on a part of the system that is especially massive and mysterious. And I thought I'd start with this recent quote from a recent review from Murray Sherman, who is really one of the forces in the field of thinking about thalamus and cortex, and in particular how they interact with one another. He wrote in this recent Nature Neuroscience review, the textbook view can be summed up as follows. Information arrives at the cortex through the thalamus in a simple machine-like relay. It's processed in this hierarchical way by King Cortex, as Gilles Laurent once called it. And then there are outputs through motor systems and other kinds of systems like that. So it's sort of up and across the cortex and then down. Of course, that completely belies the fact that there's a lot more to the system. course, yes, these are thalamic nuclei down here, cortical areas up here. This is from a fairly recent Allen Brain Institute study, building a massive database of connectivity between different parts of the mouse thalamus and cortex. These are the thalamocortical pathways, and yes, the thalamus does feed the cortex, and that part of the textbook. And I have to be careful here because disparaging textbooks is not really something I like to do. But they do give kind of short shrift to the other half of this system. These are the cortical areas, the thalamic nuclei here. And of course, the cortex talks massively back to the thalamus. And what even this picture, as complicated as is, doesn't show you is that these pathways, the numbers of pathways going down, are tremendously more common than the ones going up. So I used a thicker arrow here to imply that when people try to quantify this, there's on the order of 10 times as many descending axons from the cortex to the thalamus as there are ascending axons from thalamus to cortex. So we have this system where yes, information flows up to the cortex via these pathways, percolates around up here. The cortical areas are massively interconnected. But at the same time, each of these areas is talking back to the thalamus in massively, in quantitatively immense sets of pathways and in ways that I'm going to tell you about. So one of the stories I'm going to tell, what I'm going to try and do today is to tell you something about how these pathways work. And I'm going to lead you through some of the details, and we might get mired in some details that are more detailed than you'd care to know, but let me give you some basic anatomy here. So this is also from Murray's. Of course, Murray presents the textbook view, and then he's going to tell you how it really is, as Murray sees it at least. But I've come to appreciate his perspective on this, and this summarizes it nicely. So he distinguishes between sort of primary thalamic and cortical areas, the primary sensory areas that we think about, V1, A1, primary somatosensory cortex, and the thalamic nuclei that are associated with them. Think LGN to primary visual cortex. Things ascend this way, but then we have two pathways, two distinct, functionally and anatomically distinct pathways by which these areas of cortex talk back to the thalamus. And I'm going to talk mostly about this layer 6 pathway, but there's a parallel one from cells in layer 5 that in Murray's view, and I think it follows naturally from the anatomy, serves as a way by which one cortical area, including primary areas, talk to higher order thalamic areas, which in turn talk to higher order cortical areas. So in addition to the textbook view where things flow this way, as Murray has pointed out again and again, things also go up this way and then bounce back, up and back, and up and back. And he sees this as a pathway, as one of the pathways by which cortical areas talk to one another, as well as to the rest of the brain. In this case, this blue box here is basically all the outputs of the cortex. So I'm going to attempt at the risk of saying too much, I'm going to try to tell you 3 short stories. The first will be about corticothalamic neurons and their organization. And there I'll start in the cortex itself. Then we'll talk about the pathways as they descend into the thalamus. And I'll tell you something about the functions of these 2 different parallel systems, which turn out to operate very, very differently. And this one, if I have time, I'll talk about thalamic neurons themselves and a topic that is somewhat related to the rest, but which has really occupied my laboratory for many years, the presence of electrical synapses in the brain. And I simply can't give any kind of talk these days without having at least a little short story about electrical synapses. So we'll plunge right in. Let me give you some more background to the specific system that I'm going to talk about. First of all, we're working in mice. I'm going to talk today only about the somatosensory system. And here's a bit more detail of the circuitry involved. I'm specifically going to talk about the whisker barrel system. Should need no introduction here with Garrett and all the work he's done on this, but I'll show you some nice pictures. The primary sensory activity comes up through the lemniscal axons into the VPM nucleus of the thalamus. The relay cells of the thalamus, of course, send their axons up to the cortex. They're glutamatergic, they're excitatory, although they do engage some inhibitory processes up here. There are higher order nuclei of the thalamus associated with this, and I'm only going to talk about the posterior medial nucleus, so the prototypical higher order nucleus. It also sends excitation up to the cortex, although into different layers. So there are these parallel systems going up, and there are parallel systems coming down. So I told you about the layer 6 system. The cells of origin are in layer 6, particularly the upper part of layer 6. They come down and they do something fairly complicated where they make excitatory connections, but they also engage feed-forward inhibition, which complicates things, and I'll tell you about that. The parallel system is from cells in layer 5, and these are really the big layer 5 cells, the ones that burst, the ones that send their axons out of the cortex. These are the cells that communicate the message of the cortex to the rest of the brain. Everybody else is just sort of talking to themselves or back to the thalamus. So they send an axon down to the spinal cord, the tectum, the brainstem, all sorts of places. And in passing, a little collateral goes off and excites some of the higher order nuclei, including the posterior nucleus. What is this good for? This massive system, again, 10 times as many axons in that direction than in this direction. Bottom line is we know something, but not nearly enough, and there are a lot of competing theories for what corticothalamic systems do. Clearly, the cortex is regulating the thalamus, and thereby regulating its own input. There are ideas about how it regulates the excitability, but is it excitatory in its regulation, or is it inhibitory, or both? People have looked at receptive fields, and activating corticothalamics can change receptive fields, sometimes sharpening them. People have looked at synchrony. Activity from the cortex can sometimes be shown to synchronize activity down in the thalamus. It can switch cells. Thalamic cells are famously able to switch their excitability modes from bursting and rhythmicity to more tonic firing, and corticothalamics can have an influence on that. Rhythmicity is also something that the thalamus does very nicely, particularly because of this feedback inhibitory system from the reticular nucleus. So basically anything the thalamus does has been implicated in the effects of what the cortex, how the cortex regulates it. And I'm not really going to answer any of these questions or settle this, but what I'm going to try and do is tell you something about how these systems, what they actually do to the thalamus, and then the ultimate functions. Somebody else will figure out probably someday, soon. Garrett, you get that job. So again, barrel cortex. I'm sure nearly everybody in the room knows about it. It's part of the somatosensory system. It's a beautiful system for experimental study because the sensory map out here is it's sort of quantized by these long hairs on the face that mice and rats wiggle. There's a nice set of 5 rows of the biggest ones on the face. Those map into the pathways all the way up to the cortex. In the cortex, you can see the map literally with your eye, even with a naked eye if you've got sharp eyes. This is a cytochrome oxidase stain that I stole from somebody. So these are called barrels in the cortex. I'm going to show you a lot of pictures of barrels. But I just want to make the point here that there are actually barrels or barrel-like structures all the way down or all the way up. We start with a pattern of follicles on the face. They're matched in the first synaptic relay station in the trigeminal nuclei. Then in the thalamus, we have little barrel-like structures. They're called barreloids here, barrelets here, and barrels here. And you'll see why I'm belaboring this. But the neurons are clustered together for reasons known, well, not to any human being. The neurons that are primarily responsible for the neural activity, processing the neural activity from a particular whisker, are clustered together in these relay stations all the way up. Nearly everything I'll tell you about today is done in vitro in slices, and back in when I was still at Stanford, my very first graduate student, Eric Agmund, figured out laboriously how to cut a mouse or a rat brain in the right plane of section so that you can retain the barrel cortex here. You can see the barrels. This is just a DIC, transmitted light image. You can retain the cortex and the thalamus and a lot of the connectivity between them. And this has been a really useful prep. Studies in my lab and a number of other labs. You can see when you put a virus, an AAV down in the thalamus that is transducing, in this case, EYFP and channel rhodopsin. You can see the pathways very nicely in the slice. So we have a lot of connectivity here. Obviously, it's a slice, you lose a lot of it, but enough to really study the detailed cellular mechanisms. So I'm going to start with the layer 6 cells. Layer 6 is, again, this kind of big layer right at the bottom, full of an incredible zoo of cells. If you just look at the morphology. Again, it was one of the last layers to really get the attention of physiologists and people interested in function. It's a motley collection of pyramidal cells and the usual inhibitory interneurons. We now know that there are corticothalamic cells in massive numbers in layer 6. There's diversity among them. I'm not going to talk about that. There are other cells that are quite a large fraction of these layer 6 cells as well that don't go back to the thalamus, but instead mediate cortico-cortical connections. And then there are yet others in layer 6 doing who knows what. So I'm going to tell you in most detail about layer 6 and the pathway, the corticothalamic pathway from layer 6. One of the reasons this has not been studied much until recently is that there's no good handle on it. Until molecular genetics came along and gave us tools, there was no way to isolate these in any selective way. There is now, at least in mice, and a lot of the experiments I'm going to show you coming up in the next slides use this mouse called the NTSR1 Cre mouse. So it's a Cre expressing mouse, Cre recombinase expressing mouse, driven by the neurotensin-1 receptor gene, or promoter, for, again, reasons that aren't really relevant and are unknown anyway. This mouse expresses Cre recombinase almost exclusively in layer 6 corticothalamic cells. And this was first shown in Scanziani's lab. I think it was Hillel Desnek's work, postdoctoral work. It essentially expresses in all the layer 6 corticothalamics and in nothing else, as far as we can tell. So it's a very nice, clean Cre line. And we can use it now to express whatever we want in these cells in a very precise way. You can see this. This is just now showing you, in this case we have YFP in the layer 6 cells. So you can see the cells down here in layer 6, and heavily in upper layer 6, some of their axons and dendrites. Here, VGLU2 immunocytochemistry has been done so that you can see the barrels up here and the alignment of them all. And we use the same approach either by crossing the animals or using small viral injections to look at both the corticothalamic and the thalamocortical pathways. I just like to show these nice, colorful pictures. So here, this is work of Shane Crandall, who's a postdoc in the lab. He made an injection of an AAV into the BPM, which is the primary somatosensory nucleus. Infects the relay cells here. And what you can see in yellow, greenish yellow, are all the axons going up to the cortex. They arborize densely within the barrels, so you can see the whole barrel array. They also send their little collaterals into the thalamic reticular nucleus, which is this inhibitory nucleus that I'll tell you about in a minute. And there's another small set of deeper axons. Here's an injection into the PO that for the most part misses the VPM. So we're looking here at the higher order nucleus, somatosensory nucleus, and you can see that it also goes up to the cortex. Very different lamination. You can see it's laminating most densely just below layer 4 in the barrels, and turns out it's hitting some of these layer 6 cells. Can't see it here, but there's actually a substantial amount of input to layer 1 as well. So this is the basic architecture of the system. And let me tell you about what we think is actually, we've reinvented something that turns out Dan Simons noticed about 30 years ago. which is a sort of a new set of barrels within the cortex. And it turns out to be an organizational feature of the corticothalamic system, and that's why I'm telling you here. So, Shane, postdoc in my lab, noticed, of course, in the DIC, if you have a live slice in the chamber, you can see the barrels pretty readily. But he flipped over, he was using this NTSR animal and using various fluorescent reporters to label the corticothalamics in layer 6. And I think it's kind of dim here, but you may be able to see that there's some structure here. They're a little thin. If we had better contrast, you can see the lines between them. If we highlight the barrels and then look straight down, there are actually clusters of these layer 6 cells down in layer 6, corticothalamic cells down in layer 6, that align themselves with the barrels up in layer 4. And so Shane has now studied this in some detail, trying, first of all, to establish that there really is some kind of unitary structure to the deeper corticothalamic cells. So this is in one of the transgenics. Again, you might be able to see these blobs down here that align with the barrels in layer 4. He's measured these things, actually quantified them. These are just line scans through the fluorescence of the V-glute, which shows you where the barrels are in layer 4, and the YFP, which shows you where the layer 6 cells are down here. They obviously co-vary. Cross correlation shows very, very nice correlations and a nice periodicity, which lines up with the barrels. And to make a long story short, and then he did counts of cells. And to make a long story short, there is this sort of barrel-like structure down below in layer 6A that lines up with the barrel structure in layer 4. And there's no hint of it in the intervening area within layer 5, which is also a big, thick layer. Nothing shows up a barrel structure there. So something developmental is aligning these two. Turns out that it's the corticothalamic cells that line up with the barrels. And in fact, when Shane counted the non-corticothalamic cells, they seem to be preferentially aligned with the septa, or the areas between the barrels. He stained for some of the two major kinds of inhibitory interneurons, parvalbumin and somatostatin expressing inhibitory cells. And they show no relationship to these layer 6 barrels, so they're indifferent to this structure. Bottom line is we're proposing this as again, Dan Simons, in using very different techniques and sort of indirect methods, noticed in rats that in fact there is a substructure in layer 6. He didn't give it a name. We're going to give it a name. We're calling it infrabarrels, as in infralaminar, below the granular layers of the cortex. We've struggled with names here, and this is not perfect, but we're going with this for now. But the basic idea is that there is an organizational feature, a spatially organized set of units, or appear to be, down in layer 6A. The blue cells are the corticothalamics, and they tend to cluster more underneath the barrels in layer 4, whereas these other cells, the corticocorticals, tend to be toward the edges. We don't know what this means now, but Shane has been exploring now the connectivity between these cells and these cells, as well as between these cells and the thalamic inputs, trying to tease apart the local circuitry of the system, and have to report back to you about that. Okay, so that's sort of the site of origin, at least of the layer 6 system, that's going to be projecting down to the thalamus. So I'd like to tell you now about what that system does to the thalamus. And I like this quote from this Isaacson and Scanziani review from a few years ago, because it sort of encapsulates something that I had kicking around in my own head in a much less articulate way. But basically, synaptic excitation and inhibition in the brain are inseparable events, not just in the hippocampus or the cortex, but really everywhere in the CNS. There are very, very few places where you can get excitatory things to happen without immediately engaging, or within a millisecond or so, feed forward and feedback inhibitory circuits. And the cortex is no exception to this. The thalamus is certainly no exception. I already showed you this basic pathway. So now we're talking about the green cells here, how they come down and they directly excite relay cells, And this pathway comes from all cortical areas and goes into all thalamic nuclei. But it also does an interesting thing, which on the face of it is kind of simple, but in fact, if you think about it for a second, turns out to complicate things considerably. So it's a glutamatergic pathway. It excites the relay cells, which go back up to the cortex. But it also engages this set of GABAergic cells in the thalamic reticular nucleus. These are interesting cells because they do a couple of things functionally or circuit-wise. They feed forward inhibition. As this system comes in, they feed forward inhibition onto the relay cells. And from the same pathway, they're getting direct excitation and a disynaptic inhibition. They also get input from the thalamic axons as they go up to the cortex, so they're mediating a feedback inhibition as well. So nothing gets into or out of the thalamus without engaging this. Actually, these get into it without engaging it, but that aside, this is intrigued a lot of people, including Francis Crick, who called it what did he call it? The gatekeeper of thalamic transmission, something like that. So here's the question for the next set of slides. We're interested in this layer 6 pathway and the fact that it engages both this inhibitory feed-forward system as well as direct monosynaptic excitation. How does this shake out? And past work has been done mostly in vivo, a little bit of in vitro work. Hadn't really clarified this well. In vivo work suggested in some labs and some paradigms that you got a net excitation of cells down here. Other people said you got a net inhibition. In other cases, it's state dependent. So we wanted to sort this out. So I've told you about the anatomy or the circuitry. What's the bottom line? Does this system, when you activate in various ways, generally excite or generally inhibit? And the answer, of course, is both. What are the mechanisms? So again, we're going to use this technique with the thalamocortical slice, the NTSR animal to activate or inhibit just those layer 6 corticothalamic cells. Most of these experiments were done with viral injections to get stronger expression, as opposed to, say, crossing it with a Chalmers-Dobson floxed line. But here's an example of an injection in the cortex, labeling these cells, and you can see the tufts of axons and terminals in the thalamic reticular nucleus, the inhibitory one, and then onto the relay cells. So that's the approach. Just more colorful pictures, a little hard to see, but using 2 different viruses, we're trying very hard to understand the mapping into this reticular nucleus, which turns out to be surprisingly weird and complicated. Okay, so the first thing you might ask is, what are corticothalamic cells doing in a behaving animal? And again, there's a kind of a fairly sparse literature, but there is a literature, and it's very species dependent, it's area dependent. Some people have a very hard time finding anything that gets layer 6 cells excited, anything behaviorally, whereas other people find ways to excite them tremendously. I guess the main point I wanted to make from this work from Farron Griggs and Marty Usry in the monkey is that in the monkey and in many other species, including mice, these corticothalamic cells in layer 6 vary in their firing frequencies a lot. Sometimes they're incredibly quiet, like silent. Other times they will fire at tens of hertz up to 100 hertz in some cases. So they have a wide dynamic range, which means that the activity of their descending axons will have varying firing frequencies. So what happens when you activate these pathways? So we're going to use channel-rhodopsin expressed in the layer 6 cells selectively and in their axons and terminals to selectively excite this particular pathway. Light is usually applied down to the thalamus itself because there's enough channel-rhodopsin in these axons and terminals to allow you to activate the terminals selectively so you can actually throw the cortex away and it would work just fine. And then we'll record from usually a pair of cells of specified type. In this case, one of these inhibitory cells in the reticular nucleus and an excitatory cell in the relay nucleus. And here's what you see. So if you give a single 1 millisecond flash of light, activates these terminals, you get what was known pretty well from the anatomy in the TRN. This is the GABAergic cell. You get a very brief excitation. Here the cell has been induced to fire repetitively, and you can see that there's a brief increase in spiking frequency there. If you activate it multiple times, this is just many examples overlaid. If you now look at what's going on down in this cell, which is getting the parallel excitatory and inhibitory input, here's what you see. It's hard to see it here on this time scale, but there is again a very brief period of excitation which coincides with this monosynaptic EPSP. And then you get a profound period of inhibition. This just shuts this cell down. Here, there's really not much you can do to get it activated, and then it comes back, and this lasts for 100, 200 milliseconds. So that's what happens with a single stimulus. Of course, the brain rarely spikes once and stops, although there's a lot of sparse firing going on in the cortex. It frequently fires at much higher frequency, and that's when things get interesting. So here's the same sort of experiment, except I'm just going to show you the recordings from the relay nuclei. Here is control. The cell is just spiking along. Then we, in this case, impose a 10 hertz activation of these corticothalamic axons. And again, you see the inhibition here, pretty strong. Brief periods of excitation and mostly inhibition. And here's a raster display of that. But if you continue to fire the input, the inhibition seems to fall away, and instead is taken over with a fairly strong and kind of tonic period of excitation. So it actually switches from a period of inhibition to a sort of protracted and sustained excitation. And then we dissected in quite a bit of detail how this actually happened, because again, if you think about it, there are a lot of things that could be going on here. We only have really 3 cells. We've got that cell, and that cell. We have 2, maybe 3 kinds of synapses, that excitatory one and that excitatory one, as well as this inhibitory one. But what accounts for this switching from inhibition to excitation? It could be that something going on with the intrinsic properties of the cell. They're adapting in their spike frequency mechanisms. It could have to do with the synapses and their short-term dynamics. Some synapses depress, some facilitate. It could be that inhibition, something going on in this part of the circuitry. The inhibition is going away. NMDA receptors, modulators, the mind boggles at the possibilities. Basically, all the cellular neurophysiological mechanisms could be at play here. So we tried to tease this apart, and I'm going to give you the very quick capsule version of this. So here, relay cells were voltage clamped, and by holding at different holding potentials, you can dissect and tease apart the excitatory and the inhibitory currents in a fairly clean way, although, it's not perfect by any means. You hold it at the reversal potential for inhibition, and you can look at the EPSCs. You hold it at the reversal potential for excitation. You can see the IPSPs in a fairly pure way. You can test the dynamics of these. You can test the magnitude of them, the kinetics. And what I just want to point out a couple of things. So as you stimulate at 10 hertz, excitation, these EPSCs, facilitate. So these are strong, and it turns out these are strongly facilitating synapses. They're weak initially, but they get much stronger as you repetitively activate them. This is very common in certain pathways in the brain. The IPSCs are actually falling away and depressing. So there's a fairly prominent short-term depression here. With this experiment, you can't say for sure whether it's something in that synapse, the release, or is it something in the cell itself. And I'm going to tell you it's mainly a function of this. But so what we have is, through this train, we have ramping up of excitation while the inhibition is falling away. So this clearly would seem to underlie parts of it. Turns out there's a strong NMDA component to the excitation of these cells. Here's an experiment where we put APV on, which is an NMDA receptor antagonist, and you can see that the inhibition still falls away, but we've lost that late excitation. So maybe NMDA receptors have something to do with this. Glossing a lot of the details, I'm just going to get you to the punchline here. So We use a technique called dynamic clamp, which some of you I'm sure are familiar with. Many of you may not. You know about current clamp and voltage clamp. Dynamic clamp is a way, it's been around for a good 20 years now. The technique, as computing has gotten cheaper and faster, it's become easier to do this and do it well. But basically, it's kind of a hybrid system where you record from a neuron in current clamp. You allow it to fire the way it wants to fire, but then you simulate conductances, and you can program in whatever kinds of conductances you want. You control their size, their kinetics, their dynamics. So you can mimic synaptic inputs or any other kind of ionic current in the cells. And that's what we've done here. So these are real experiments you record from a cell, but you use rapid feedback and calculate and simulate the synaptic inputs to the cell with the computer and feedback through the electrode. So we figured out enough about these currents and the conductances that underlie them to simulate them. The AMPA component of the excitation looks like that. The NMDA currents are generating a lot more charge, and they look like that. Both are facilitating. The inhibition is very strong but depresses with time. So am I supposed to be using that? Can't hear it? I can wear both. Can't hear me in the back. Oh, I'm sorry. All right. How's that? Is that better? Hear me now? Give me a sign, somebody. Yes, no, sort of. Better. All right. Try to make it even better. Okay, so this is a way of really testing our hypotheses. What's involved here? Which components of the synaptic properties are essential for getting this switching? So here we are at control conditions. We've simulated the two forms of excitation and inhibition, and it recreates in the real cell something very similar to what you see when you actually activate the natural synapses. Now we can play with this in ways that you can't. You don't necessarily have a drug to do all the things you'd like to do. So we remove the AMPA receptor component just in the excitation onto the relay cells. And you still see the switching. You lose that very fast excitation, not surprisingly, because that's what generates the really rapid triggering of action potentials. And we lose, surprisingly, a lot of the late tonic excitation. So AMPA turns out to be important, even though it's not generating a whole lot of charge there. NMDA receptors are profoundly important for the late excitation. That wasn't a big surprise, again, because they carry so much charge and they're being ramped up because of this facilitation. Here you can do some things that you don't have drugs to do. You can play with the properties, the dynamics of the system. So again, control up here. Here we take away the facilitation in the excitatory synapses under the cells, and it completely blows away that late excitation, so it never really switches over into excitation. Here we take away the depression of the inhibition, and you can see that the inhibition stays on longer. There's still late excitation, but still a lot of inhibition. And there are a lot of similar sorts of approaches that I'm not going to show you. So bottom line, what causes this switching of the corticothalamic layer 6 system from inhibition to excitation? It's complicated, multifaceted. Facilitation in that excitatory synapse, the fact that there's a lot of NMDA current here is important in these same synapses. It turns out, I haven't shown you, there's facilitation at this synapse as well. That plays a minor role. It turns out that the intrinsic properties here, the burstiness of these cells actually also causes them to spike less, but that doesn't actually change the effect on this synapse. It's primarily the depression of the inhibition that leads to this loss of inhibition in the beginning. So it's the combination of largely depression of the inhibition, facilitation of the NMDA receptors and the AMPA receptors that sort of switch it as it comes on. Now this is all done with very unnatural tetanic stimuli. wanted to try something slightly more natural, short of going to an in vivo prep, which we're working on now. And we used a technique that, again, came out of some work that Adesnik did in Scanziani's lab. Annabelle, this is what I was referring to earlier. So it turns out that in the cortex, you can induce gamma rhythms, or at least gamma frequency network-dependent events, really robustly and beautifully, in a perversely sort of unnatural way, I have to say. But you can induce cortical gamma by putting channel rhodopsin into pyramidal cells, or the excitatory cells. In our lab, it works in almost any layer in a whole variety of different areas. Put channel rhodopsin, in this case, in the layer 6 pyramidal cells driving the corticothalamics. The stimulus is a light ramp, so it's a kind of ramping up of light or even a steady pulse of light, something that is just sort of driving the cells in a steady way, not necessarily a very strong way. But when you excite groups of pyramidal cells via feedback inhibitory loops, they induce gamma frequency excitation. So here's an LFP in the cortex, a field potential. Here's a voltage clamped cell down in the thalamus. These are very tightly coordinated. I won't show you the cells up in the cortex, but they're also tightly coordinated. There's a nice gamma frequency oscillation going on here. Fairly high frequency in this case, almost 80 hertz, although it's dependent upon how strongly you drive the system. It has a lot of the features of natural gamma, except that it is sort of perversely periodic and strong. But we did this up in the cortex. Now the light is shined on the layer 6 cells to induce this cortical gamma. And we looked at what we got down here as we activate this layer 6 system via the gamma frequency oscillations. And the bottom line is that you basically recreate what we saw with pulses. This is in voltage clamp. These are inhibitory currents. So you see the switching here. It starts off mostly inhibiting the cells. Here it is in current clamp. And then with time, it actually activates the late sustained excitation. So the basic phenomenon is recreated with a somewhat more natural stimulus pattern up in the cortex here, and via presumably the same basic mechanisms. I should have said, if you have any questions, stop me now. Okay. I don't have much to say about in vivo experiments, except that this is something we're working on now. So far, just in anesthetized animals, we're just beginning now to make recordings in unanesthetized animals. Always a challenge. And the recordings we want to make are whole cell recordings down in the thalamus in particular, as well as the cortex. So fairly deep structures. You can actually make very nice recordings, as many people have shown these days. When you activate the layer 6 corticothalamic system, you get very nice inhibition, brief excitation. There's suggestion under inhibition, under anesthesia here, that of this switching, but we haven't yet really recreated, or maybe it doesn't work in vivo. I don't know. That's a basic question that we're asking. We'll be exploring these mechanisms. And then, of course, what you can do here is use some natural stimuli. What we can get is very nice. If we activate the layer 6 corticothalamic pathway briefly, you can get very nice suppression of whisker-evoked activity down in the thalamus. So the mechanics of this are roughly working now, and something we're working on. Okay. So I think in the interest of time, I'm going to keep this down to two short stories. This one is fairly brief. I'm going to tell you about this other parallel corticothalamic pathway, the layer 5 pathway. Just to remind you, it's this one up here. Starts with the big output cells of the cortex, these big pyramidal cells in deep layer 5. They send their axons down to other parts of the brain. And, oh, by the way, throw off a collateral here to excite relay cells only down in the higher order thalamic nuclei. Again, how do we do this? It's a little trickier because we don't have an NTSR-like animal for the layer 5 corticothalamics. So we're using a couple of different alternative strategies that are so far working pretty well. One of them is to use a virus, a Cre-off virus. So we want to express channel rhodopsin in the layer 5 cells but not in the layer 6 corticothalamic cells, because we want to look at the layer 5 pathway selectively. So you can engineer, we didn't do this, we got it from another lab, but you can engineer a virus that expresses in the absence of Cre recombinase. So it won't express in the red cells here, the corticothalamics in layer 6, but it will express in the pyramidal cells nearby, including the layer 5 corticothalamics. This actually shows you that, although it's hard, I'm sure it's hard for you to see. This is down sort of in the top part of layer 6. It is kind of cool. The red cells are all layer 6 corticothalamics. Then you see other cells that don't have red in them, so they're not layer 6 corticothalamics, but they do have beautiful rings of reporter expression in their somatic membrane. So they're expressing channel rhodopsin, and then you can go in and patch these and show that they have nice channel rhodopsin current. So we have a pretty good handle on the layer 5 pathway using this technique. And then you can go down in the thalamus and look closely at these, and it reveals a really interesting feature which we didn't discover. It's been known for a long time, but it's shown nicely here. I don't know if you can see the red speckles. So we're looking down in the PO nucleus, where the terminals are. The red speckles are the terminals of the layer 6 system. They're very dense, they're very small, and this tells you something about and these are the terminal boutons that release glutamate. The big yellow globs are the terminals of the layer 5 corticothalamics. Same nucleus, so they're overlapping. They've got parallel pathways into PO. But these, other people have done EM on these, they're huge glutamatergic terminals onto the same cell. So the layer 5 system makes these massively large, they're in some cases 3 or 4 microns in diameter, excitatory synapses onto the cells. Whereas the layer 6 system is making these tiny little, very dense glutamatergic synapses. And you can do an experiment. I'm not going to show you a whole lot of data today, but actually this was done with a different strategy. So there are lines of mice that express Cre only in layer 5 cells in more or less good ways. This is something called the RBP4 line that we've been using. Only has Cre in layer 5 cells. You can put a virus in here, get channel rhodopsin expression. I like this picture because it shows beautifully that these layer 5 axons, as I told you, they don't arborize in the TRN, so that you don't expect them to engage the inhibitory feed-forward pathway. They don't arborize in VPM, the primary nucleus, somatosensory nucleus down there. They do arborize in PO, so they are going selectively to PO as they should. And what happens when you activate these? You record down here and you activate these terminals. What you get is, first of all, no inhibition, to speak. You can't see any inhibition here. What you do get are very fast and sort of massively large, even with single axon stimulation, very, very large, fast excitatory currents onto the relay cells. And instead of showing the switching or facilitation that the other system shows in here, it actually shows a very strong depression. So this is what Murray Sherman would call a driver pathway, as opposed to the others, which he would call a modulator pathway. There's a lot of freight with those words, but it does sort of capture the essence of it. So again, to come back to Murray's picture, put it together, and to summarize, I think we can, if we consider these two parallel pathways by which the cortex regulates the thalamus, the layer 6 pathway is, again, massive and ubiquitous and goes to all of the nuclei. I don't know what its function is, but it does seem to have a fairly powerful influence and an ability to regulate it and activate, at low frequencies at least, fairly profound inhibition onto the cells down here. Although when jazzed up in the cortex at higher frequencies, these cells will have the potential at least to switch over into something much more excitatory. And it could be that they are, in Murray's terms, more or less modulating in a bidirectional way the ability of the thalamus to transfer information upward. The layer 5 system's doing something completely different. And here I'll just lean on, so Murray's whole point in this review is mainly to talk about this layer 5 system. So it's, there's some interesting features here, right? It only goes to the higher order nuclei. These are the cells that are also, and mainly probably, sending their axons out to the output structures, many of the motor structures, the corticospinal, corticotectals, corticopontines, et cetera, to actually drive behavior. And at the same time, they're informing these higher order nuclei of what's going on. So you can't, there's no more efficient way of telling another part of the brain what one part of the brain is doing than by having a bifurcating axon that sends the same basic message. So there's kind of an efference copy, and he likes to think of these now as efferent copies that go into the higher order nuclei. What are those nuclei doing? They're going back up to the cortex, and often to yet higher order areas of the cortex. So this does seem to mediate a potentially fast and powerful pathway from one cortical area to another. Whether that's actually the case and what they're doing isn't entirely clear. All right. So I'm going to skip my last story, unfortunately. Sorry, let me just go back and do it this way. And I just want to wrap up. So, just to summarize, and I'll tell you the bottom line of the story I skipped. First of all, I think it's interesting to think about this new organizational structure to the corticothalamics. Whether we'll see that in any other system besides the barrel system, which has this unique sort of structural organization, remains to be seen. But it does suggest an interesting specialized circuitry there that we're working on. We looked in some detail at how the layer 6 system regulates the cortex, and it has this bidirectional ability, frequency-dependent ability to either suppress or excite the excitability of relay cells. What I didn't tell you is about gap junctions, which are near and dear to my heart. Here's the short story. They're all over the brain, and we should deal with that and figure out what they're doing there. In the forebrain, cortex and thalamus, striatum for that matter, they are, in the mature animal, exclusively interconnecting inhibitory cells. The evidence that they connect in excitatory cells is, let's just call it weak. It's mainly inhibitory cells that are talking to one another through gap junctions. Gap junctions are channels that interconnect two cells. They're very simple, biophysically simple channels that allow current to flow back and forth in both directions. They're very good at helping cells coordinate their activity, but can also mediate a lot of more interesting kinds of dynamics. It turns out that probably every inhibitory neuron in the cortex makes gap junctions to other inhibitory neurons, but for the most part, they do it in a very cell-type selective way. In the thalamus, these thalamic reticular cells, which are all GABAergic, are also coupled by gap junctions. And what I was going to tell you, if I had more time, I'd tell you about a new story we have where in a model of tuberous sclerosis, which is a genetic disorder that involves seizures and certain OCD symptoms and autism often, we're finding that there are ectopic electrical synapses in the excitatory cells, in the relay cells, that seem to be able to mediate synchrony that it might underlie some of the abnormal EEG rhythms and even the seizures in these animals. But that's for another day. I'll just tell you the people who did most of the work. Most of what I showed you was the work of Shane Crandall and Scott Cruikshank, who my long-time collaborator in the lab. Scott and Shane is a postdoc in the lab. Tim and Sung Chan were grad students who did a lot of work on the electrical coupling that you didn't get to hear about. Rosa and Liz have been working on the tuberous sclerosis and other thalamus-related projects. And I thank you for your attention and happy to take questions. Tim.
That was great. So how would the switching be used? I mean, it looks as though the inhibitory period is just a couple spikes, and once that's done...
Well, yeah, I mean, the inhibitory period depends on frequency. I only showed you here one frequency, 10 hertz. But you know, of course, it varies with the frequency at which you hit them. If you go down to low frequencies, all you see is inhibition, and that inhibition lasts a couple hundred milliseconds. So you can get a more sustained kind of period, quite a prolonged period of inhibition if you keep the frequency low.
It's not just time dependent, it's frequency.
It's frequency dependent, yes, very frequency dependent.
Okay, and those inhibitory interneurons in a lot of other systems are perfect for setting timing.
Uh-huh.
They're great at establishing periodicities.
Yeah.
So,
Yeah, we haven't really looked carefully at that kind of thing here. I mean, again, when they're firing at low frequency, well, I have no doubt that they'd be pretty good at mediating timing signals. But
whole network's
and I
coupled.
yeah, well, and right. I didn't point it out, but when we did induce gamma in these, you can see it's kind of the nature of the thalamus, been much studied by Dave McCormick and others, that because of the feedback, largely because of the feedback inhibition through the reticular nucleus, there's a kind of resonance, an inherent rhythmicity to this thing because of the feedback part of this limb. You know, thalamic neurons like to intrinsically burst at certain frequencies, relatively low frequencies. If you play with the system a little bit, you can get it to mediate a feedback inhibitory rhythmicity. There's some strong evidence that this is actually that this feedback inhibition plus the burstiness of the thalamic cells underlies absence seizures, which are about 3 Hertz seizures. And you can actually see that here. So the drive here is a pretty steady high frequency gamma, but in the beginning, while the inhibition is still strong, it sort of falls into this fairly slow rhythmicity. So that's, you know, so at that time, at that level of timing, it's definitely mediating fairly synchronized and time-locked rhythms. But at higher frequencies, again, I didn't show you, but if you induce that high frequency gamma in the cortex, you can actually see very nice input to the cells down in the thalamus at gamma frequencies. So, and there's other in vivo evidence that there might be. Is that me?
There are 2 questions at Emory.
2 questions from the same person.
I don't know.
Okay. Shoot.
Go ahead, Emory.
We have about 15 folks here, all love the Thalamus. This is Dieter Jäger. I have 2 quick questions. One is the NMDA component of the driver synapses. How much NMDA is in those?
How much NMDA? A lot of NMDA is measured by the current. Hi, Dieter. You want numbers? I could show you a picture. You know, of course, it's dependent upon how strongly you drive it and everything else. But much more NMDA current than AMPA current. I don't know if that's enough for you, but I could send you the specs because we had to, you know, program this all up in the dynamic clamp, so we needed real quantitative information.
But this was the layer 6, right? I was interested in the layer 5.
Oh, layer 5. I don't have the answer for that. Just to judge by the kinetics of the currents, it looks like it's mostly AMPA mediated, but that we haven't. That's really still in progress. We haven't really teased it apart that way. Sorry.
Just a more general question. We were very interested in the motor thalamus, VA, VL over here. Any basic comments how similar or dissimilar the VA, VM nuclei would be in terms of layer 5 and layer 6 input?
Yeah. I would guess, we haven't studied on either of them. We have been interested in their thalamocortical effects, and you know they have interesting trajectories up to the cortex, but we haven't looked at corticothalamics into them. They have the same basic corticothalamic anatomy for layer 6 and layer 5, so I would expect to see some very similar effects, but it'd be worth doing.
So maybe in interest of time, just one more
Yes.
I'm not a Cortex aficionado, so, but just a general sort of broad question, and maybe the answer to this specific system. You alluded to the fact that there were many of these cortical neurons that were controlling or feeding that thalamus or regulating it. First of all, just like order of magnitude, how many are we talking about?
How many?
And then the general question is, why so dang many?
Yeah.
Is it just
Well, that was.
repeated many times, or is it?
So I can't answer the first question, but I could look it up. On the second question, yeah, that was one of the premises here. Why, there's this massive system that and it dwarfs the ones that are going up that we all talk about and that the textbooks dwell on. Well, again, I could go down the list of things people have suggested. I like the idea that there's a modulation going on, and this system seems like an interesting one. It's not a modulator in the sense that you would throw norepinephrine or acetylcholine onto a set of cells and get some kind of slow, protracted shift in their excitability. This thing is actually it's bidirectional and regulated fairly quickly by the firing frequency of the layer 6 cells. But I would expect it to have a kind of general modulatory effect on the excitability of the cells, although, again, it can mediate more rapid timing of things as well. Other people have emphasized more the rapid timing possibilities and how that affects receptive field properties. These cells are so-called relaying information up to the cortex. They have receptive fields of their own that change when they get up to the cortex. So this is modifying the sharpening them, some people have suggested. Changing the receptive field properties in fairly significant ways. I like the idea of Murray Sherman about the layer 5 system, this driver system being a kind of efference copy. That is informing these higher order areas of cortex via cortico-thalamo-cortical connections of cortical output, motor output, driving those cells in a way that's quite different from what this system does. They're very sharp, largely probably AMPA-mediated, precise timing, strong, quick synapses that could excite those cells and send a fairly reliable signal back up in short order.
Why don't we thank Barry again for a great seminar.
Thanks.